Hello and welcome to Lost in Science. We are your sturdy Lost in Science crew sailing in the good ship of whatever this is, through the seas of knowledge, through the oceans of imagination and wonder, shall we say. Is that overselling things a little bit? I don't think so. Joining me this week, we have Lyndon. Hello, Lyndon.
Hello, Captain, my captain. Good
I
to
don't know
see
that I'm the
you.
captain. I'm see myself more as a Gilligan kind of character. And we have Stu.
Hi, Chris. How are you,
Hi.
Skipper?
Yeah, very good, very
Gilligan.
good. Now I understand, Stu, that you have a interview for us this week.
I do have an interview. I was reading up on vaccines because there's a couple of different things going on with people not keeping up with vaccines. One of them is to do with measles, which is mainly in the United States, which doesn't really apply to us. The other one's with diphtheria, which does apply to us and is having a resurgence in some parts of the country. Obviously, the research on COVID vaccines in particular is ongoing because there's still some things we don't know about them. We do know they were safe and effective. We do know a lot about them. There's still boost programs going on and people still getting them. So I wanted to talk to someone who has been researching that very topic. So I caught up with Associate Professor Claire Von Mollendorf, who is from the Murdoch Children's Research Institute. Her background is in paediatrics. She started off researching pneumococcal illness and got drafted into COVID research along with everyone else at the time. But some of that research is ongoing. So they've been looking at the long-term impacts of the vaccines and the effectiveness of the vaccines and the longevity of the immunity that the vaccines provide to people. And there's a lot of interesting stuff in there. If you want to know, in shorthand, you have better health outcomes getting a COVID vaccine than catching COVID from a wild type virus. That's the long and short of it in the research, and it is ongoing. So there's more results being collected all the time from various cohorts in different countries. It's really interesting stuff, but I just thought it was, it was a timely reminder that the protection you get from vaccines does fade over time, and I wanted to ask her all about that.
Great. So you're giving it a bit of a boost, I suppose. But it's always good to inject some science into the debate, I suppose. I'm going to stop jabbering away and let Stu have a shot. So yeah, let's just get on with the show.
Yes, I see. Good news, everyone!
You're listening to Lost in Science.
So it's been a while since we had a story about vaccines on the show, and we did talk a lot about them kind of 5 years ago for obvious reasons. But I've got with me on the show this week Associate Professor Claire Von Mollendorf from the Murdoch Children's Research Institute, who's been doing some research on vaccines and their effectiveness. And I just wanted to get her on the show this week to talk about some of the things around vaccines. Thanks for joining us, Claire.
Thanks for having me, Stuart. I'm really happy to be here and to share some of our data. So I'm an epidemiologist working at the Murdoch Children's Research Institute. So I work mainly in infectious diseases and vaccine research. And we do some work in Australia, but also more broadly in the Asia Pacific region as well.
And what is your focus on epidemiology? Or are you broadly across epidemiology, or do you focus on specific diseases, or what's your main focus?
So I mainly focus on pneumococcal disease. So the pneumococcus is the most important bacteria causing pneumonia and meningitis in children. And we've got a really effective vaccine called the pneumococcal conjugate vaccine that's been introduced in children over the last 2 decades. So that's really my focus is predominantly in children, but during the pandemic, obviously there was a need for more COVID vaccine studies. And so our group was involved in 3 big clinical trials in Mongolia, Indonesia, and in Australia.
On COVID vaccines specifically?
On COVID vaccines specifically, yes.
Okay. So, I mean, one question I have been asked, and I think I answer it reasonably well, but one of the things people ask is, why are there so many different COVID vaccines? And there's dozens around the world. And the question is why? Yeah.
Exactly. So that's a great question. So because this was a global pandemic, we realized that really early we needed to vaccinate everybody everywhere in the world to make an impact and to end the pandemic. So that meant that we needed a lot of diverse vaccine candidates, so lots of different vaccines that could be produced at scale through different manufacturers and supply chains, and that would work across a range of populations. So for example, a lot of the vaccines, like the mRNA vaccines, really needed ultra cold storage, and that wouldn't be a good vaccine, for example, to use in rural Papua New Guinea, where you had to, use a cooler box. So we needed a lot of different vaccines. We also needed vaccines that were affordable. So if vaccines were really expensive, obviously governments wouldn't be able to afford that. And then I guess lastly, countries wanted assurance that they would have access to vaccines. So early in the pandemic, obviously, countries were producing their vaccines and stockpiling them. So countries wanted their own vaccines and produced their own vaccines because that would give them assurance that they would have access to it. And the reason why the vaccines were different, I guess, is that a lot of countries had different expertise in different vaccine platforms. So the vaccine platforms have been used for other vaccines. And so some countries or labs or scientists had different expertise. And so they pursued those types of vaccines in their production. And I guess as well, in terms of the mRNA vaccines, there were certain platforms. So mRNA platform where you can change the vaccine really quickly. So a lot of the old, I don't want to go into too many technical, but for some older vaccines, you need to kind of grow it in eggs and the whole process takes really long. Whereas the mRNA technology, you can alter and change the vaccine really quickly. So yeah, that's the reason why there were so many candidates. And lastly, not all vaccines that were developed actually got to market. So for example, when we started, there were over 200 candidates that were actually in being researched during the pandemic. And WHO only pre-qualified 12, and FDA and the European Union actually only recognized about 10, and then in different countries there were only about 50. So we really needed a lot of vaccines to, I guess, get success at the end.
And was one of the reasons that you mentioned that there was 200 candidates and then there was a dozen were sort of recognized. Was the effectiveness of the vaccine part of that recognition? Did they say, let's pick the best ones out of the 200?
Exactly. So as you know, there's a really robust, long pathway that vaccines go through to get to market. And the COVID vaccines had to follow the same pathway. It was just accelerated. So all vaccines first get checked in the lab. They then go through animal testing, and if they're safe in animal testing, they then go into human trials. And there's 3 phases. So phase 1, you basically checking that there's a good immune response to the vaccine and that it's safe. And then phase 2 is slightly bigger trials. And then phase 3 is when you have those huge trials where you're checking if the vaccine works. And then during COVID, there were kind of set parameters by which they were checking if the vaccine worked. So against severe disease, for example. And then then needs to go through the FDA or the European Medicines Association for it to be approved and go into use in market. And a lot of people, I guess, were worried because they heard that these vaccines got emergency approval, but it was still a robust process. The manufacturers still had to show that the vaccine was safe, that it wasn't causing any harmful side effects. So it still followed this really robust pathway, I guess, to show that it was effective, that it was safe before it was used. As I mentioned, obviously, the development was speeded up, and I guess that worried a lot of people, but that was only because scientists around the entire world were working together for once. So everybody was like, we need to solve this problem. They kind of, instead of usually normal vaccine development, you finish one stage and go to the next stage. But what they did, especially, I'm sure you know about the US Warp Speed project, they collapsed all the stages. So they did a lot of stages concurrently instead of successively. And for example, when the US found that there were really strong candidates, so like the Moderna product, they started producing the vaccine before the trials were finished. So they really compressed the timelines. But that doesn't mean that there wasn't the same checks and balances in place. And the manufacturers still had to continue the trials and report back to governments. And we all knew scientists knew what was happening at the time. Everybody was watching the literature, seeing what the vaccines were doing. So yes, that's how we knew that the vaccines would work. And now we know that they work because we've vaccinated billions of people. And when we started vaccinating people, people stopped dying. So pre-vaccines, we all saw those terrible videos of people in ICUs dying. Once we started vaccinating people, that stopped. So I guess that's the best data we have that the vaccines actually worked.
Yeah, that's a real world test, even though they had been extensively tested.
Exactly.
So one of the things with COVID vaccines particularly is the idea of getting boosters, and this is related to dosage and also related to how rapidly the virus mutates, which is something that happens with most viruses. They do mutate over time, but the COVID virus seems to mutate quite quickly. And so, I mean, obviously, that's a good reason to vaccinate people against it anyway, is that's how it mutates, is in people's bodies. But how do they figure out the dosage and how often boosters needed and all of those things? How are they figured out? We know that it works, but how do we know how much we need and how often we need it and why do we need it more often and all those things? How's that worked out?
So I guess, first of all, the first question is, how do we decide what dosage to use? So when we do clinical trials, the manufacturers use dose escalation techniques. So what they do is they have different dosages in the trial and they look at immune responses in people. And then when it goes to big phase 3 clinical trials, they try different doses and see which one works against the disease. However, in COVID, because they were speeding up the process, they couldn't try lots of different doses. So what they did is they tended to choose the highest dose to be the highest dose that didn't cause any safety or bad side effects. So for example, when we started, the Moderna dose was 100 micrograms. But then as the studies progressed, they actually reduced that to 50 micrograms and they saw that 50 micrograms worked. So that's the usual project, is that they tried the highest dose just to be sure, but then it often decreases over time. So your first doses, you might need a higher dose, and then for boosters, you might need lower doses, because your primary doses is the first time you've seen the virus, or the first time you've seen the antigen.
Yeah.
Whereas for booster doses, you've seen it before. So you're just kind of reminding your immune system.
If one National Science Week activity is not enough for you, please head along to Howler on Thursday the 20th of August for Sci-Fight, the science comedy debate. This National Science Week, the theme is The Internet Can Be Saved. Watch 6 scientists and comedians battle it out to argue whether the internet is worth saving or whether we should just go out and touch grass. Sci-Fight, tickets available now. The battle takes place on Thursday the 20th of August at Howler Nightclub in Brunswick. You've tuned into Lost in Science, where Stu is talking to Associate Professor Claire Von Mollendorf about COVID vaccines.
As I mentioned, we had 3 clinical trials. So 2 of our clinical trials in Mongolia and Indonesia were looking at that question about boosters. In both those trials, we were looking at whether you could give half the dose of boosters and that would produce good enough immune responses. And the reason for trying half doses is there was a shortage of vaccines. So countries were producing their vaccines and stockpiling them. And usually lower, poorer countries that weren't making their own vaccines couldn't get them. So we needed some way to make sure that we could boost as many people as possible. So in those studies, we found that the half doses overall worked as well as the full doses. So, for example, in the Mongolia study, over 24 months, so up to 2 years, we found that using a half dose was as good as a full dose. And what our studies did, which not all studies do, is that we didn't just look at the antibody responses. So we also looked at the cellular responses. So cellular immunity, it's, I guess, specialized white cells in your body that are also, when you get exposed to an infection or vaccine, these white cells respond to that. And what those, what the cellular immunity tells us is it shows us protection. It's a good proxy, I guess, for protection against severe infection. But it's also the part of your immune system that shows memory as well. So in our studies, we looked at both antibody responses and cellular immunity, and we saw that it was strong for both the half and full doses. But what we did see with those trials and our trial in Australia, I guess, is that there's initially there's very good immunity up to 6 months, and then it does start to wane. So that's why if you look at the booster recommendations for high-risk groups, it's usually every 6 months. So for the elderly, for severely immunocompromised groups, they usually recommend boosters every 6 months, and for other groups every year. And we saw that in our data in terms of waning of immunity.
So that's really interesting because it basically allows people, or allows the responsible authorities to double their doses of vaccines by halving the amount that they give to each person. So that massive money saving, I guess, as well as helping more people.
And it's also, I guess, good. So fractional doses are not unique to COVID vaccines. It has been used for other vaccines as well. So, for example, for yellow fever vaccine. So it's very good strategy for any vaccines to get, as you say, greater coverage for the same cost.
Yeah, and I guess, that if there's criticisms about the COVID response, cost has been one of the ones that comes up all the time. So that's good that we can reduce that. Is there any way potentially in the future to extend the immune response beyond 6 months, so that maybe we'd only need an annual booster, or is that something people are working on?
In terms of new vaccines, I guess they have been looking at different vaccines. I think for COVID, unfortunately, because it's no longer a priority, a lot of funding that was there was cut. And so I do know that Pfizer were looking at different types of vaccines and different dosages and stuff, but I'm not sure if that's still ongoing because a lot of the interest has kind of run out. And with the change in government in the US, where most of the funding was coming from, that might have petered out. But they were looking at extending, at different formulations to extend the immune response. But hopefully it'll still be important for later later pandemics or that. But I guess in terms of long-lasting protection, so our third trial was in Australia, so that was slightly different from the trials in Mongolia and Indonesia. So there we enrolled healthy adults who had already received 3 doses of vaccine. So they'd had their 2 primary doses and their first booster, and we were really looking at second boosters to look at protection long-term against infection. And what was good about that trial is that we randomized adults to receive a Moderna mRNA vaccine and a Novavax vaccine. And then we also had a control group. So the control group was self-selected because we didn't want to detect to people that they couldn't receive the vaccine. So we had a group of individuals who didn't want to receive additional vaccines, so they didn't get a 4th dose, and we followed them up over 12 months. So the current data that we just published is over 12 months, but we've actually followed them up further to 30 months, and we're busy looking at that data. So hopefully late in the year, that data will be available. But what we saw that up to 12 months, so initially, the antibody response to the mRNA vaccines is higher than the protein vaccine. So that's the Novavax vaccine. But over 12 months, the cellular response, so your white blood cell response in the 2 groups is very similar. So we know that overall your immunity is the same between the 2 groups. And then what we saw is that both the vaccine groups had a much better antibody response than the control group. So your level of immunity was high in the vaccine groups to the control group. So we know that getting a vaccine is better than just getting ongoing infections. And what we saw is that in the first 6 months, both vaccines had over a 50% higher efficacy against infections than in the control group, but then that waned over time to 12 months. So as you're saying, it's kind of your immune response is strong in the first 6 months. But it was still better to get a vaccine than not. But what was good, I guess, in this group where they've already been vaccinated and now they're just getting boosters, all the infections that we saw were mild. So there was nobody that had hospitalization, even in the groups that weren't getting ongoing boosters. So we know that getting a vaccine as a booster is better than not. But even if you don't, because you've had your primary schedule of vaccines, if you're getting infections, they're going to be mild.
Yeah, I mean, this is what we've all been hoping for, and that's exactly what you're finding in the research as well. I think we'll have to wrap it up now, but thank you for joining us, Claire. It's really interesting stuff, and it just goes to show the ongoing investigation of the scientific method. You have to keep checking and checking and checking, and we get better and better information.
And hopefully when we have our 30-month results and late in the year, early next year, we can come back and keep people interested in getting boosting and continuing to, I guess, support the science.
Yes, we'll check back in with that when that gets published. Thanks for joining us on the show, Claire Von Mollendorf.
Thanks so much, Stuart. Thank you.
And that is all we have time for this week on Lost in Science. Thank you for joining us in Getting Lost. If you have any questions or suggestions for the team, get in touch with us by email. We are lostinsci@gmail.com. You can send cheap tweets to us at lostinscience1 on Twitter, or you can find us on the ubiquitous Facebook. Lost in Science is recorded at the studios of 3CR in Melbourne on the land of the Kulin Nation and is broadcast across Australia on the Community Radio Network with the assistance of the Community Broadcasting Foundation. You can find a podcast version of the show on 3cr.org.au, or you can tune in the way you did this week when we return in our usual time slot to get
Lost in Science!
Thanks for listening to a 3CR podcast. 3CR is an independent community radio station based in Melbourne, Australia. We need your financial support to keep going. Go to www.3cr.org.au for more information and to donate online.